Thank you all for coming today. I am really excited to talk about my dissertation research on examining the convergent evolution of craniofacial morphologies in weakly electric fishes. First, I want to back up and talk about how we define convergent evolution. The textbook definition is independent evolution of similar traits or adaptations in different clades. And this is seen across all vertebrates, including examples like flight in birds and bats, the kind of pokey little hairs seen in hedgehogs and echidnas, but there are also a multitude of examples in teleost fishes as well. And when we think about convergent evolution, we really need to examine it from multiple different viewpoints, including genetics, development, morphology, and function. But in my work, I'm going to focus on a couple of groups of teleost fishes and a couple of very specific morphologies to determine if we can quantify convergent trait evolution. Now, my two groups of interest include Apternodidae, a group of electric fish from South America in the order Gymnotiformes. There are about 100 valid species, and they're found in the humid neotropics, including Central and South America. These fish are kind of funky looking. They're not your typical fishy shape. They have a very elongated anal fin, but no dorsal fin. And their electric organ is actually so long that it spans across their entire body. They have a high frequency wave type electric discharge and are the fastest known biological oscillators. Compared to Mormyridae, this is a family within the order Osteoglossiformes, which is found in the humid Afrotropics. There are over 200 species in this family, and unlike Apternodidae, these particular fish have a dorsal, anal, and caudal fin present. They have a point source electric organ that's found in their caudal peduncle, and it does not span the entirety of the body, and it produces a low frequency pulse type electric signal. So we're already seeing some differences, but some convergences as well between these particular families of interest. And my methods are going to be examining craniofacial convergence between these two groups. And I'll be doing this using CT scanning at Friday Harbor Labs, and then I used open source software called 3D Slicer to segment out the skull and take a look at some 3D geometric morphometrics, including 22 homologous landmarks across the skull. And the next couple of slides show us some of the diversity in head shape that we see within these two different families. In Apternodidae, I was able to sample 43 species with 160 individuals. And for Mormyridae, I sampled 41 species with 229 individuals. And so moving forward in my methods, I used R packages, geomorph, and PhiTools to perform a Procrustes superimposition to standardize the coordinates, a generalized principal components analysis to identify axes of shape variation, and then I performed a maximum likelihood ancestral state reconstruction. So to look at some of our results when we compare all of the specimens within both families, here is the principal components analysis that I was able to uncover with these methods. PC1 is seen on the x-axis and PC2 is seen on the y-axis. In pink, we have the Mormyridae specimens, and in green is Apternodidae. Now we see some pretty distinct kind of separation between these two groups. There's kind of one little area where they seem to possibly overlap or be pretty similar in shape, but the rest of the specimens are pretty distinctly in separate groups here. If we want to visualize some of the morphologies that we're seeing across these two axes, here are some of the skulls that I have reconstructed. So over here on the left-hand side, on the extreme negative PC1 axis, we have Sternoncorhynchus, which is in the Apternodidae group. On the other side of PC1, we have some of the short-snouted species of both Mormyridae and Apternodidae. And looking at PC2 extreme values, we have short-snouted species like Petrocephalus, like Portergus down here versus Campylomyrus up here on the top of this particular graph. And in order to visualize this even further in terms of our landmark data, I did trust network visualizations in order to look at the differences in these extreme values of these axes. And so what we see with this particular visualization are actually two of the most commonly seen shape trends when we visualize shape change in gnathostomes. On PC1, we have the phenomenon of heterocephaly. So this is the relative size of the snout or the face in the brain case. And this is variation along the dolichocephalic and brachycephalic axis. On the left-hand side, we have the dolichocephalic phenotypes with an elongated snout and a contraction of the brain case. Versus on the right-hand side, we have a foreshortening of the snout and a relative expansion of the brain case. And this phenomenon has been documented in gymnotiforms previously. Corey Evans, in his paper in 2017, found this phenomenon across multiple species within this order. And this is a visualization of one such species, Compsorius samuli, where we actually see heterocephaly happening within the same species, typically through ontogeny. And this phenomenon is seen across different vertebrates and gnathostomes, and is seen both in ontogeny and also across phylogenetics, like we're seeing within this family itself. The second shape trend that we see in this principal components analysis is heterorhynchi, which is seeming to load on PC2. And this is the relative flexion of the neurocranium, and it's often associated with mouth position and dietary or feeding strategies. So on the top of this extreme axis here, we have species like Campylomyrus with an elongated downturned snout, versus the other side of the axis, we have Portergus species or Apternotus species that have a shorter snout that seems to be more terminal. But there are actually some other interesting phenotypes that we can see along this axis. And this is one example found within gymnotiforms. In the genus Sternarchella, we have sister species Sternarchella sima and Sternarchella ducus. And Sternarchella sima actually has more of a terminal, almost subterminal mouth, and is a benthic invertivore, versus its sister species, Sternarchella ducus, that actually has a dorsally pointed snout. So this particular species eats the scales and tails off of other fish and has a snout that's pointed pretty much directly upwards. When we break it down into analyses just within each of the families, first I'm going to take a look at Apternotidae. And I again did a principal components analysis, and we can see some of the extreme phenotypes along this particular graph in this figure here. On the left-hand side, again, we have the very short-snouted species. An example would be Odontosternarchus or Portergus. On the right hand, we have the species that are very long-snouted or elongated snouts like Sternarchorhynchus. So again, we're seeing the phenomenon of heterocephaly being loaded on PC1, and we're actually also seeing the same phenomenon of heterorhynchi on PC2 when we compare species like Sternarchorhynchus with their downturned snout versus species like Apternotus that have more of that terminal mouth at the very end of its snout. And what we see is actually kind of an inverse relationship when we visualize PC1 and PC2, where the higher we get PC1 values, we actually seem to get a lower PC2. And this is indicating that as the snout is getting longer, it actually tends to point more downwards, which is kind of an interesting phenomenon. And this is again visualized when we look at the trust network visualizations for both PC1 and PC2, where we see the phenomenons of heterocephaly and heterorhynchi coming out pretty clearly. I then performed an ancestral state reconstruction on just Apternotidae based on Max Bernst's tree from 2019. And what we found was an estimated ancestral condition of an intermediate length rostrum. And this is based on continuous traits of PC1 values. We also found polyphyletic origins for both dolichocephaly and brachycephaly phenotypes, and more instances of change towards those brachycephalic phenotypes than the longer snout dolichocephalic ones. And so this is indicating that it's potentially easier to break developmental pathways and prevent growth, kind of get that shorter snout phenotype, than it is to make or lengthen developmental pathways and continue to get those longer elongated phenotypes. When we break down the results into just more Myridae, again, I did another principal components analysis, and we see the same overall trends for PC1 and PC2, with heterocephaly and heterorhynchi loading very, very heavily. Also, we can somewhat see in some of the genera that same inverse relationship between PC1 and PC2, but it doesn't seem to be quite as clear in these particular fish. And so again, with that trust network visualization, we see heterocephaly being on PC1 and heterorhynchi being on PC2, with some of the extreme examples shown here. The ancestral state reconstruction again showed a potential estimated ancestor of an intermediate length rostrum, multiple instances of evolution towards short-snouted phenotypes, and at least one towards longer snouted phenotypes, as we saw in the Aptenotidae family as well. And so when we take a look at this overall, what we find is that despite superficial appearances of convergence, like these two fish that are visualized over here on this slide, craniofacial shape underlying those external morphologies may not be as convergent as previously assumed. But we are seeing some similarities between the groups in terms of their potential ancestral state and the number of times that we're seeing extreme phenotypes evolving in both lineages. And with that, I would like to thank my committee members, lab mates, funding sources, people who offered and let me borrow specimens, friends, family, and of course, the fish. Thank you.